My goal for coming to college was to do medical research, and I knew that being in the College of Medicine, right surrounded by all these hospitals, was the best way for me to get right into a lab right away, start researching and taking individualized classes that were geared towards research.
So for my research, I worked in the neurology department, and my research was more about understanding the properties of creatine and how a lack of it would lead to schizophrenia or bipolar disorder. So I did more of my research towards stem cells, and I enjoyed it. I enjoyed working with it, understanding the different properties of being in research.
What I do is in diabetic wound care. So diabetics, their wounds do not heal at the normal rate that humans' wounds heal, or normal humans' wounds heal. And because of that, it leaves susceptibility to infection. In order to solve that, what you can buy, or what the majority of medicine does at this point, is they apply dressings to the wound. Now that dressing is usually filled with silver, because silver is a very good antimicrobial and antibacterial agent. So the dressing itself prevents a lot of bacteria from getting in, and the silver inside the dressing clears up the bacteria that's already in the wound. Now the issue that happens throughout all of this is that the companies that produce these dressings, they don't say specifically how much silver they put in their dressings. I wanted to test exactly how much silver are in these dressings. So far in the last 2 years, we've collected a lot of data. We've learned, and we use a machine called an ICP-MS, which tests for parts per billion of silver ion and particle, which is very, very, very small amounts of silver. And what we do is we took 3 different types of dressings. So we're able to see kind of the disparity in terms of how much silver ion and particle are really in these dressings.
This past summer, I spent my summer researching at the Pasteur Institute in Paris. So I was doing microbiology research, researching Klebsiella pneumoniae and the type 2 secretion system. So I was working in the lab every day and experiencing the work life of France and learning more about science.
For research, it really just taught me about being determined and always just moving forward and just trying not to give up. And I believe that determination that I learned in research really helped me as an individual.
I've been to a few conferences to share the work that I've been doing, and hopefully we'll get some publications in there soon.
And I'm really thankful to have this opportunity and be part of this amazing structure and program and everything.
I like the individualized part of it. I get to be me and my research, but I see it as a different creative process. I like that challenge and the many failures in order to succeed. That's just something that I see as a different problem-solving method, and I enjoy trying to tackle those challenges.